Hello, everyone. All right, today we're getting into week 2 of embryology. This is implantation and differentiation. So we got a lot to get through, so let's get going. So implantation is what we start with. Week 1 is the development of the zygote into the blastocyst. So that's where we're at right now. We're at the blastocyst, which the blastocyst has two types of cells, the embryoblasts and the trophoblasts. Down here, the trophoblasts. These, in week 2 of development, these are going to divide further into two different things, which we'll go over in a sec. So it's kind of like the week of 2s, which kind of helps memory. But that's what we have. We have a blastocyst. It has a blastocele, an open space. But regardless, this happens around day 6 to 8. 6 to 8, mainly, we're going to say around 7. When that happens. What happens in implantation is the blastocyst travels all along the uterine tubes up until, we're going to call this the uterus. Yeah. Not an artist. But it's going to come out here and say this is the endometrial lining. It's going to implant right here, or wherever in the endometrial lining, and implant into the functional layer, functionalis of the endometrium. That's endometrial layer, or a point arrow, that's going to become the decidua. What the decidua is, it's simply the endometrial lining that has been changed and altered to allow more nutrients to exchange into the blastocyst or the embryo in order to develop it further. That's really what it is. But that's the implantation part. It's aided by syncytiotrophoblasts, which we'll discuss in a second. That kind of helps it burrow into the endometrial lining and create a space in it. But we'll get to that. So that's the implantation process. Now we're going to get to differentiation. So I'm going to erase all this. Okay, differentiation. So remember, it's kind of the week of 2s, if you will. So we'll go through that. So remember our two types of cells that we have in the blastocyst are the trophoblasts and the embryoblasts. So like I said earlier, these two differentiate into two things. Trophoblasts, and for not deciding that I don't want to write the massive word, I'm just going to put its prefix. So these differentiate into syncytiotrophoblasts. So I'm just going to write syncytio, and then cytotrophoblast. That's where those two go. The embryoblasts over here differentiate into epiblasts. I'll write the whole thing. Epiblasts and hypoblasts. So let's go over what the heck these are. So easy one over here. The epiblasts, these two form the bilinear disc, by the way. Linear disc. All right. And actually, I decided I want to start over here. So the cytotrophoblasts are really just a layer of cells. So they're kind of just there, and they'll differentiate further later down the road, but we're not worried about those guys right now. The syncytiotrophoblasts are really important. They secrete enzymes that allow for burrowing. So like we had in the implantation, when the blastocyst had implanted into the endometrium, that was aided by syncytiotrophoblasts that secreted these enzymes, allowing it to kind of burrow in, kind of like some animals that you could probably name. But it also secretes HCG, which is human chorionic gonadotropin hormone. When that's super key because we need this hormone to keep the corpus luteum alive. If you don't know what the corpus luteum is, go look at my video or someone else's video about the uterine cycle. But the corpus luteum helps the embryo get to full stage, full development. And so we need this HCG to keep that corpus luteum alive. So that's what the syncytiotrophoblasts do, and we'll see these guys further down the road too. The cytotrophoblasts are kind of there. Then over here we have the bilinear disc, which is formed out of epiblasts and the hypoblasts. So know also this: the epiblasts are the primitive version of the ectoderm. So I'm just going to write ectoderm down here. And then the hypoblasts are the endoderm. Okay. All right. So what do these guys kind of do? The epiblast will eventually form the amnion. There's a process there, but we're not going to go over that process. And then the hypoblasts eventually form the yolk sac. All right. Both of these are kind of membranous-like things. The amnion is kind of is near the endometrial lining side of the embryo, and the yolk sac is more on the outside. But they both form kind of cavities. There's an amniotic cavity, and the yolk sac is a cavity, but they have different functions. So we're gonna go over that. So the amnion over here, what does it do? The amnion, I'm gonna make some analogies here, so be ready. But one, it is a shock absorber. So kind of like the things on cars that prevent you from feeling every single bump in the road. Same thing with babies. We don't want babies having bumps. Shock absorber. Two, it's also a humidifier. What I mean by that is it keeps the cells moist, keeps it water, keeps them and not necessarily humid, but they have liquid. So that's kind of important. It's also kind of a scrape prevention. And yeah, that's a lot of what the amniotic fluid does. It's mainly from the maternal side. The maternal side creates this amniotic fluid. Eventually, when the embryo formed more, there's some pee in there from the baby. But this is the stuff, amniotic fluid. That's the stuff that releases when a pregnant woman says their water broke. Same thing. So that's for there, and I think there's a few more other functions, but we're not going to go over that. The yolk sac, what does that do? The yolk sac has a good amount of functions, so be ready. So one, they have primordial germ cells. These things are going to become the gonads and the gametes and differentiate further, so they're pretty important. The most important is it's the site of nutrient exchange, which we need nutrients. That's the thing we need most, nutrients, so we can grow. Also, will eventually form the GI tract. It'll also lead to the formation of the EEM. The EEM is the extra embryonic. I don't know if you can see this. The extra embryonic mesoderm. So as you can see, this is kind of the mesoderm part, and then here we have the ectoderm and the endoderm. So you can kind of see how those relate. It also is the site of original hematopoiesis. So we're going to get some blood cells going. And it also has some shock absorbance. And I almost forgot over here, what it also does is a pretty key thing of what the amnion does is it's a thermostat, if you will, keeps the temperature in regulation, which is pretty key. We want a good temperature and we want it to be protected. We also want the yolk sac to have nutrients and do all this stuff. So those are some of the functions of the amnion and yolk sac. There may be more, but so that's all in day 8, by the way. So I should probably put that. That's all day 8. So moving on then. Let's see. Sure, I'm going to move you just a little bit. We're going to go to this side of the screen a little bit. Now we're going to go to day 9 and 12. So day 9 and 12, this is kind of when the maternal sinusoids kind of come into play. The maternal sinusoids are simply just the mom's capillaries that have been expanded, enlarged, so that more nutrient exchange can occur. That happens through lacunar networks that are networks of lacunae, lacunae being spaces. And that is kind of accelerated, done by these syncytiotrophoblasts right here. Those guys are pretty crucial. They're very invasive, and that's pretty important. So that's the maternal sinusoids. They help with nutrient, blood exchange, stuff like that. Not blood exchange, but nutrient exchange. But then to around day 12, this is when the EEN, then it'll eventually form into the EEC, the extra embryonic cilium, but for right now it's the extra embryonic mesoderm. So that forms and kind of connects amnion and the yolk sac, and then the chorion forms. chorion forms from the syncytiotrophoblasts, the cytotrophoblasts, and the EEM. So that's all the chorion. And the chorion is huge because it continues to secrete HCG. It also does a good amount of other things, but we won't go into that right now because I'm running out of time. And then finally, day 14 is when the connecting stalk comes into play. Connecting stalk is simply just the primitive form of the umbilical cord. So, and that is the last day of week 2. So obviously this is kind of just a quick overview. There's much more. The chorion does more. There's more to the EEM that we could go over, but that's for a later video or later session. But remember, so this is implantation and differentiation of week 2. Okay, then we go into week 3 and then a lot more happens there. But thanks for listening. That is week 2 of embryonic development.